Welcome back to the medical water and soil microbiology practical lecture 6. This is Jihan University The Hague and the Faculty of Health Sciences Medical Microbiology Department, the fourth stage. Today our topic is detection of fungi from soil sample. So if you remember, we previously had some topics and subject about fungi in general. So mycology, if you remember, and then fungal isolation and fungal infection, of course. The fungi spread in all of the environments, especially soil and air, the spore in the air and water, but particularly in the soil. So and the spores spread to the air. So how can we isolate? This is in our practical topic. How can we isolate the fungi from the soil, and then how can we detect this and do the identification to those fungi which we isolated? If you know that we have two main types of fungi, molds and yeast. So yeast is unicellular, the oval or round in shape, especially reproduced by sexually by budding. By budding. And then the molds, multicellular and sexually and asexually reproduced in general. And we have the main thing that molds is multicellular and produce a lot of pigments, and the spores and the pigments which they produce are toxic, of course. These are some types of medium of fungi which isolated on the media. And the main general and specific media for fungi is SDA, if you remember. So we very talked about and explained about SDA, Saburo agar, and we have more than this, but this is which we have worked in, of course. Another shapes of yeast from soil, and look, it is if you see here the shape and the colony, the shape of colony on the media. We have, of course, the variety of shapes and colors, especially in molds. In our practical, so we need, of course, media and petri dishes, slides, cover slides, pipettes, and test tube. We need SDA media and multi-agar media. So those both of media we're gonna to work in and do the inoculation and isolation on both of these media. Microscope, of course, and we need the staining solution, which is lactophenol cotton blue. And this is all of this, we have it, and we will work in hand. Of course, you will work in hand in together in the lab. So you can simply have 1 gram of soil sample. So some of you will bring the soil sample as an activity, of course, to a 9 milli of distilled water, and then do mix, and then do the serial dilution method, so to around uh, 6 or 8, 6 or 8, it is okay. 10, I mean, sorry, a serial dilution method. And if the last one, I mean, if it is 6, so the fifth tube and sixth tube, or it is 8, if it is 8, it is better to do 8 dilution method and fold. So the seventh and eighth tube, we will have 0.5 milli or 1 milli is okay, because we need the fungi, a lot of fungi, I mean molds isolated from the soil, spread, inoculated 1 milli from the last two tubes on SDA agar and on malt agar. So SDA agar and malt agar, of course, we can then incubate in 28 centigrade in the incubator for 3 to 6 days. It's okay. You can observe daily and see if it is grow, and then in the next we will do the staining, of course, on microscope by lactophenol, of course, and observe, of course, examine the formed colonies and recognize so the color and morphology. Uh, after the incubation, of course, you can have a small colony on the glass slide, and in a drop of water and a drop of lactophenol, cotton blue, lactophenol, you have it, of course, and put a cover slide, and then immediately examine under microscope in 10 and 40, 10x and 40x, of course. You will find the colony I'm going to show you now. For the yeast slide, you can have one colony, of course, from the petri dishes, petri dish, and then on the glass slide and cover slide, of course. You can here we can use both lactophenol and crystal violet as well, and then examine under microscope. So you can find like this. This is the yeast, and the yeast under microscope by lactophenol cotton blue. Thank you for your listening, and this is our practical session.